Hi everyone, I will be discussing applications of normative models to schizophrenia to hopefully highlight the utility of normative models to examine heterogeneity of cortical abnormalities in schizophrenia, as well as the biological basis of this heterogeneity. But to provide a brief background, one of the most frequently reported MRI findings in schizophrenia is altered brain morphology, and this is frequently characterized by case control losses in cortical thickness, and this measure describes the distance between the grey and white matter interface and the grey matter C-sep interface as shown here. So while this finding of reduced cortical thickness in schizophrenia is quite robust, recent normative modelling studies, including ours, indicate substantial heterogeneity across individuals diagnosed with schizophrenia. And this is both in terms of the specific loci affected and in terms of the severity of cortical thickness alterations. So one of the interesting questions that might arise is what contributes to this heterogeneity? One possibility, which we hypothesize, is that there are differences across patients in the cell type specific contributions to their pathological profile of cortical thickness. So for example, perhaps some patients show predominantly glial-related pathologies, while others show more extensive neuron-specific pathology. Here is one example of recent molecular evidence for cell type specific features in the pathology of schizophrenia as well as in other neuropsychiatric disorders. And this study, very briefly, aimed to place transcriptomic changes evident in schizophrenia and other neuropsychiatric disorders into a systems level context. To this end, they performed a weighted correlation network analysis to create independent networks of genes and isoforms implicated in the differential expression of these disorders. And this figure here just shows a hierarchical clustering of these co-expression networks where the x-axis reflects the gene modules. Then they assessed relationships between each of these modules with markers of all major CNS cell types. And what I want to point out is that nearly half of the genal isoform modules demonstrate significant cell type enrichments, which is demarcated with an asterisk. Another important feature to note is that all major CNS cell types are implicated in these transcriptional modules, suggesting that all major CNS cell types are involved in schizophrenia. So in other evidence, postmortem studies highlight molecular changes that characterize patient subsets, for example, in inflammation-related transcripts. So each diagnosis here is split between a high and low inflammatory subgroup. So the high group is shown in a darker color. And as you can see, more schizophrenia patients are falling into this high inflammatory subgroup relative to bipolar patients and controls. So a follow-up study found that brains from this patient subset showed alterations in molecules important for cellular processes. For example, the high inflammation schizophrenia subgroup displayed higher, as you can see here, here it is, sorry, intercellular adhesion molecule 1. And they also found in this study that this was enriched in brain endothelial cells and astrocytes. And this might suggest that some individuals with schizophrenia display greater involvement of these particular cell types in their pathological profile. So just to summarize here, this should highlight the importance of cell type specific changes in the molecular pathology that characterizes schizophrenia, as well as particular patient subsets that might be characterized by particular cellular processes and, for example, inflammation-related transcripts. So based on this previous evidence, we aim to determine whether cortical thickness heterogeneity relates to differences in neural cell type involvement as inferred from established gene expression data from the Allen Human Brain Atlas. And this resource includes a high spatial resolution atlas of gene expression across the brain for over 15,000 genes. And the basic idea for this study is that if we have a map of gene expression for each cell type, and we have a map of cortical abnormalities for each subject, which are both registered to the same atlas, we can see whether patients' brain abnormalities are spatially aligned to the regional gene expression for a particular cell type. So basically, you can perform a correlation between the deviations in each region with the gene expression level of a particular cell type in each region. So the study participants here were drawn from 3 independent data sets: the Human Connectome young adult sample, which was combined with the Human Connectome project for early psychosis, and a validation data set was drawn from the Australian Schizophrenia Research Bank. Here is a brief methodological overview. As I just mentioned, we used a discovery and validation data set, and to characterize cortical thickness heterogeneity, we applied normative models of cortical thickness variation in each brain region, which were established on healthy individuals, and then patients were referenced to these percentiles for each brain region. And then we obtained these spatially resolved gene expression data for particular cell types in the brain. And this was done previously by Jacob Seidlitz and colleagues, and these data are publicly available at this GitHub link. So to do this in brief, they used microarray gene expression data from the 6 adult human donors provided by the Allen Human Brain Atlas. And then these gene probes were mapped to the Desikan-Kiliani Atlas, which yielded a matrix of genes by brain region, where each element here reflects gene expression level. These were compiled into particular gene sets. And these groups of genes or gene sets were compiled from all available large-scale single-cell sequencing studies in human brain tissue that have basically identified cell type or cell subtype marker genes. So this information was used to group the genes according to CNS cell class. We then calculated the mean expression of each cell type gene set in each of the cortical regions within the MRI registered ABA dataset. And this resulted in 7 transcriptional maps of major cell classes within the central nervous system. So now first I will describe the results from normative modeling here. So these cortical renderings at the top display the mean deviation score for each region across all individuals with schizophrenia, respectively for the HCP and ASRB data sets. And as you can see, infranormal or lower than normal deviations were more severe in frontal, temporal and insular cortices, and were most severe in the insular and rostral middle frontal cortex and the caudal middle frontal gyrus. In terms of normative modeling, here are percentile curves, for example, brain regions where the red curve represents the 95th percentile and the blue curve represents the 5th percentile. And individual deviations from this normative range were then determined for each individual with schizophrenia. And those falling below the normative range are highlighted here in red. If you repeat this method for each brain region, what we can do is yield an abnormality map for each individual. An example is shown here for a male who's 31 years of age. And what you can see in particular is the deviations in each brain region, which can be thresholded to highlight just those regions that are falling below the normative range for this particular individual. This plot on the right shows results from normative modeling, and specifically the portion of patients and controls with infra or supranormal deviations respectively. And what it shows is that fewer than 20% of the individuals diagnosed with schizophrenia showed significant deviations for any given brain region. And this means that more than 80% of patients are actually falling within the normative range established for each region. And therefore, collectively, this demonstrates that, similar to previous work, regional variation in cortical grey matter abnormalities shows substantial variation among individuals with schizophrenia. And our hypothesis here is that this heterogeneity may reflect distinct cellular sources underlying cortical thickness alterations across individuals with schizophrenia. This figure here displays the distribution of correlation coefficients between each individual's cortical abnormalities and interregional gene expression of a particular cell type. And so the values range from -1 to 1. The black curve represents the null distribution of correlation coefficients, and the observed correlation coefficients in patients are shown in red respectively for each cohort. As you can see, patients are showing more dispersed patterns of coefficients relative to the null distribution, and this is consistent with our hypothesis that associations between cell type gene expression and an individual's cortical abnormalities would only be evident in a subset of individuals, not in all the individuals with schizophrenia. So if I just zoom in on a particular plot here to highlight what we're doing, the blue color shows the null hypothesis, which is the distribution of correlations that you would expect to see by chance alone. And this can be contrasted to the observed correlation distribution across patients, which is shown in red. And this means that regional gene expression across genes that encode for excitatory neurons correlate more strongly with cortical abnormalities in schizophrenia than you would expect by chance alone. And collectively, these findings illustrate that the spatial patterning of cortical thickness deviations relates significantly to expression gradients of genes that regulate specific cell classes, and these include astrocytes, endothelial cells, not microglia, but oligodendrocyte progenitors and neuronal cell classes as well. Okay, we next wanted to move beyond a group level analysis to characterize heterogeneity in the degree to which each patient's cortical abnormality profile correlates with interregional cell-specific gene expression profiles. So to this end, these person-specific correlations, so each element of this matrix is reflecting a correlation between each subject's cortical abnormality profile and gene expression of a particular cell type. This was fed into unsupervised clustering, which yielded 3 broad patient subtypes based on the gap criterion. One of the subtypes was characterized by predominantly associations with neuronal and endothelial gene expression, which consisted of 22% of patients. A larger group were characterized by associations with mixed cell types, and a third group predominantly by astrocytes and oligodendrocyte progenitor cells. These portions are only displayed here for the ASRB cohort, but were fairly consistent with the HCP cohort as well. And this broad cell class stratification wasn't explained by demographic or clinical factors. So as shown here, subtypes 1 and 2 were characterized by more widespread thinning in contrast to subtype 3, which showed comparatively milder cortical thinning. A similar pattern also emerged at the whole brain level, whereby mean cortical thickness deviations computed across the entire brain were lowest in individuals comprising subtype 1. So now that we have these subgroups based on heterogeneity in cortical thickness and coupling of these abnormality profiles with cell type specific gene expression scores, we wondered whether this grouping was at all valid. So to test this, we used genotype data from a subset of patients with available genotype data in the validation cohort. And the motivation for this is that cortical thickness alterations and cortical thickness in general is in part heritable and relatively stable across the life course, both in individuals with schizophrenia and healthy individuals, and especially relative to other MRI-derived phenotypes. And this suggests that genetic risk factors must be to some extent responsible for phenotypic cortical thickness alterations in schizophrenia subjects. We therefore aim to validate cell-based patient subtypes with person-specific polygenic data that captures genetic variation linked to changes in each neural cell type. Specifically, to clarify, we computed schizophrenia polygenic risk scores that were constrained to particular genes encoding for particular cell types. So in the validation analysis, what we found was that if we just take individuals comprising subtype 1, which was that neuronal and endothelial subtype, higher polygenic neuron scores in this subtype significantly covaried with lower mean cortical thickness deviation scores. Importantly, this association was not observed in patients comprising subtype 2 or subtype 3, confirming the specificity of this result to individuals residing in the neuronal endothelial subtype. We observed this in other subtypes as well, but I don't have time to go through all of this, so I'll just select particular examples. So although the 3 subtypes provided the optimal clustering solution, they were not distinctly separate, as you may have seen on the previous slide, suggesting that characteristics of cell-based patient subtypes are not mutually exclusive. Rather, individuals differ in the degree to which they exhibit cell type linked deviations in their cortical thickness profile. Therefore, we additionally validated subtypes based on what we called a soft cluster approach, whereby individual membership of each subtype was characterized continuously from 0 to 1. For example, soft subtype 1 shows that the R value decreases as more subjects with low membership scores were excluded from this correlation analysis. And this effect was repeated in subtypes 2 and 3, suggesting that the severity of cell-linked cortical thickness deviations co-vary with a portion of genetic liability ascribed to matched cell types. Okay, so just to summarize here, decomposing cortical thickness heterogeneity through combining normative models of brain phenotypes with multi-scale omics enables perhaps prioritization of particular schizophrenia subtypes, as well as particular cell types of interest for future disease modeling efforts. So in brief, these normative models can be leveraged to probe biological and genetic mechanisms that drive heterogeneity. In this study, 2 independent cohorts were classified into cell-based subtypes based on cortical thickness deviations from normative models, combined with spatially resolved gene expression data. And collectively, these studies call into question the viability of ongoing endeavours to derive group average cortical maps of schizophrenia pathology, and suggest that while schizophrenia is a multicellular disorder, the composition and degree of cell type specific involvement probably differs across individuals. I would like to acknowledge all the authors involved in this study. They all played major roles in contributing to these analysis and the conception of the study idea. And I would also like to acknowledge the participants who volunteered their time in these studies, and thank you to the funding bodies that support this work. Thank you.